Jonathan Wyndham is a Clemson Extension Specialist stationed at the PD Research and Education Center. And Jonathan, today you're going to try to make some sense of plant breeding, especially as it comes under this umbrella term of genetically modified organisms.
Right, and so there's a big disconnect between the general public and the scientific community over vocabulary, and a lot of the times it seems like semantics are splitting hairs, but there are differences between these terms, and so I want to clear up some of those differences today.
Well, let's start with just how do we form an organism? What are the building blocks?
All right, so for the sake of this talk, we're going to stick with atoms as being the smallest unit of matter for us.
Thank you.
Okay. And so an atom, viewers at home might be familiar with things like a hydrogen atom or a helium atom or oxygen atom. Atoms are the smallest unit of matter for the sake of this talk. So when you have a group of atoms, they come together to form molecules. And so you've heard of molecules before. Water is an example of a molecule. Caffeine is a molecule. Sucrose is a molecule. A molecule is just a group of atoms. It's as simple as we're going to get. So there are different types of molecules. And in genetics, there's one type of molecule that we talk about a lot, and that's called a nucleotide.
Oh, okay.
And we use 4 letters to represent the 4 nucleotides. There's a 5th one, but we're not going to get into that right now. But we talk about A, T, C, and G. And so if you've ever seen Jurassic Park, you'll see these letters all over the movie, A, T, C, and G. And so these are just certain molecules in genetics that we talk about. So when these 4 molecules come together, they create a larger molecule that everyone should be familiar with by now, called DNA. And DNA stands for deoxyribonucleic acid. You don't need to know that. You just need to know it's a big molecule made of these 4 letters, repeating over and over in different order. So within DNA, we have certain sections of DNA. And these sections we refer to as genes. And genes code for a certain product, and those products lead to traits of an organism. So the color of a cardinal, or the flower color of a rose, or the height of a corn plant, these are controlled by genes, okay? So all of your DNA together, or your DNA, when it bundles itself together, creates these things called chromosomes. A chromosome is a bundle of DNA. You have multiple chromosomes. Living organisms usually have multiple chromosomes. And when you take all of the chromosomes together into consideration, you get something called the genome. And the genome is all of an organism's DNA. So it's like if you looked at a set of encyclopedias. A chromosome would be an individual book in that set. The entire set together would be your genome. That's all of the DNA that makes an organism. And then that's how we go from an atom to an organism.
All right.
Yeah.
Okay. And we're interested in how things reproduce and combine. And so let's talk about reproduction.
Okay, so yeah, let's talk about reproduction.
All right.
Okay. So you can have asexual reproduction or sexual reproduction. And asexual reproduction, something every gardener is familiar with. So asexual reproduction is reproducing plants via cuttings, divisions, suckers, and runners. Those all lead to, it's a form of asexual reproduction. And when I say asexual, it yields a plant that has the exact same DNA as the mother plant. So if the mother plant has this DNA sequence, any cuttings or divisions that you take from that mother plant will also have that exact same DNA.
And we call that vegetative reproduction sometimes too,
Vegetative,
is that right?
clonal, because it has the same DNA.
All right.
The other side of this is sexual reproduction, and this is when you have an organism that's created from 2 different sets of DNA. And sexual reproduction is what animals do, and plants as well. But people are most familiar with sexual reproduction in animals. So in sexual reproduction, we have one parent's genome here, their DNA here, and the other parent's. These 2 will reproduce sexually to yield an offspring that has a DNA sequence composed from half of the mother and half of the father. So I'm a hybrid between my parents, you are a hybrid between your parents. You have half your mother's DNA and half your father's DNA. That's sexual reproduction.
And we get different traits can be expressed depending on how this genome gets
Shuffled.
shuffled.
And so that's the great thing about sexual reproduction is that it leads to variety and diversity. And so with the clonal propagation, you just get the same old every generation. Sexual reproduction, you get shuffling of genes, you get new combinations of genes, and that's how you get diversity, which is very helpful for plants because if you're living in a rough landscape without being cared for, you need a diverse set of genes to help you survive.
So if one year there's a drought,
Right.
some of the plants are going to be more adapted to staying
will.
alive. And if you have a wet year, some of the plants might have more adaptations that let them live through
Right.
wet seasons.
And so you get those adaptations through sexual reproduction,
Okay.
and that's the variety that helps a species survive.
And then for us, because we enjoy our food so much, that's how we get different tasting foods.
Right. So, like with roses, I've been on the show before talking about rose breeding. When I breed roses, I select a mother plant and a father plant, and then I cross them to create a hybrid between those two. And when I do that, I'm looking for a specific trait to come through from recombination between these two parents.
And our plant people sometimes are looking for disease resistance
Right.
or flavor or other
Fruit
qualities.
size, drought tolerance, salt tolerance, new traits.
So breeding is extraordinarily important.
It is, very much so, and we've been doing it for hundreds of years. Most of our crops have been bred in some form.
Should we talk then about how methods used in plant breeding? You say they're conventional breeding
Yes.
ways, and then we've got some that are genetically
Sort of
engineered.
newer, yes.
Okay.
Yeah, let's do that.
So I guess we'll start with conventional.
Okay, so conventional breeding, that includes things like we mentioned, cross-pollination, and that's probably the oldest breeding technique because that's the one most available to us. Cross-pollination is just the transfer of pollen from one plant, a male plant, to a female plant. That yields a hybrid, a sexual offspring, and this offspring will be half of the mother and half of the father. Cross-pollination, plain and simple. Another conventional breeding technique that we have employed is called mutation breeding. And this is when you expose a lot of seeds or a lot of cuttings to radiation or chemical mutagens. And so when you do this, you're hoping that the DNA changes in these cells or in these seeds or cuttings, and you're hoping that whatever that change is, it might be beneficial. Since you're just kind of throwing the seeds in the radiation, it's going to be random, but that mutation might be good. That comes from that.
So you might plant 500 seeds, and 2 of them might have something interesting
Right.
in the new plants.
And so this is a very common breeding technique. Any crop has probably been, at one point, someone's attempted mutation breeding with these crops.
And since it's random, it's considered conventional plant breeding.
It is a conventional plant breeding method.
All right, okay. And is there another type of conventional plant breeding?
So there's a thing that we can do with plants called chromosome doubling. So let's say we have a plant cell, and plants cells are normally square-ish because they have their cell wall. But in the plant cell, they have DNA. And when they go to divide, normally, let's say this plant has 24 chromosomes. Okay, that means when it divides, it will produce 2 cells, each with 24 chromosomes. Because
Alright.
the mother cells have to produce identical daughter cells for the plant to survive. And so when this happens, the amount of DNA in this plant cell has to double so that each daughter cell gets the same amount. So for a brief moment in the cell, you will actually have 48 chromosomes. Then those split to either side of the cell and are deposited in the daughter cells. Sometimes things can go wrong naturally, or you can induce them. The 48 don't split evenly. So when that happens, you get one daughter cell that has 48 and one that has none.
Whoa.
And that's chromosome doubling. You are doubling the amount of chromosomes in a daughter cell of a plant. And so when you do that, that typically makes the plants bigger because there is physically more DNA in that cell. So the cell has to be larger, which means the plant as a whole is typically larger. Thicker leaves, thicker petals, larger fruit.
And this can happen sometimes in nature, and we
Yes.
can also sometimes encourage it to happen.
Yes, so it can be a spontaneous error in cell division, or you can apply radiation or a chemical mutagen to induce this.
All right. And that's also considered conventional, and it happens sometimes just in nature.
It does.
There are some methods of plant breeding, though, that are considered kind of human-driven.
Yes.
So let's talk about some of those.
So we refer to these as genetic engineering. And so with genetic engineering, it's done in a lab and it's man-made. And so one of the oldest genetic engineering techniques that we employ, we actually learned from nature. There is a soil-dwelling bacteria called Agrobacterium. Yes, and so this is a naturally occurring soil-dwelling bacteria that naturally infects plant cells to modify the DNA of these plant cells.
Wow.
Okay, so scientists realized they can hijack this bacterium and add their own DNA or whatever sequence they wanted into this bacterium, and then that bacterium would inject that into the plant's genome. And so that's one of our oldest methods of genetic engineering is you take a plant, you dip it in a vat of agrobacterium, and hopefully you get the change that you're looking for.
And you said that this agrobacterium naturally kind of goes into plants. Did you tell me something about sweet potatoes?
So yes, sweet potatoes, so we've actually found some of this bacterial DNA in sweet potatoes because they were infected in the wild. I mean, we didn't do it, the bacteria did it themselves. So this renders sweet potatoes naturally transgenic. Yes.
This is complicated.
Yeah, very much
But we
so.
can use it to select a trait and try to put it into a plant.
Yes,
Using this.
agrobacterium.
Wow.
Yes, and
That's
so
pretty cool.
It is, and it's really neat.
And then gene editing, you
Yes.
you say you kind of use scissors to do that.
So that's sort of the newest technique. You'll hear the word CRISPR in the news a lot, and CRISPR is the newest thing, it's gene editing. And with CRISPR, what's really important with CRISPR is that let's say you have a stretch of DNA. And so I've drawn DNA as a double helix. But let's take that double helix and we're going to uncurl it for a minute, just so you can see.
All right.
And we're going to lay it on its side. So imagine you've got a stretch of DNA, and this contains your gene that you want to edit. With CRISPR, what I can do is I can come in to this DNA, I can tell CRISPR, I want you to go here, and I want you to create something called a double-stranded break. So CRISPR will come and
Whoa.
break the DNA. When that happens, that triggers the cells to fix this because this is a very big issue. You can't have your DNA, your recipe book, split in half. So when this happens, the cell has two options. It can either repair this DNA using a template, which you can provide, or you can let the cell repair the DNA on its own. And when that happens, normally you'll get mutation. So if you provide a template, you can insert your own sequence here with CRISPR. Or,
Hopefully another trait that's desirable.
hopefully, yes. Or if you let the cell repair itself, then a lot of the times, the insert you get won't be as large as the template you provided, but it won't be exactly the same as it used to be. And when that happens, you can silence a gene because you've broken the gene. So that's the benefits of CRISPR is precision genetic engineering. And so that's the big push now in agriculture is using CRISPR because it's so precise. Unlike the agrobacterium we talked about, you can supply this CRISPR complex with something called a guide RNA. And all that means is that we know the sequence of this DNA, those A, T, C, and Gs, we know the order they're in. So we can tell the guide RNA, okay, throughout the whole genome, I need you to find that one spot that has this sequence. And once it finds that spot, I want you to make that break. And that's the benefit.
Because it could be something that was causing something undesirable or harmful to the plant or the organism.
And so I mentioned the word mutation. A mutation is a change in a DNA sequence. And there are different mutations that occur. You can have a few nucleotides deleted. You can have some added. There's something called an inversion where the DNA breaks out, flips around, and puts itself back in. Lots of bad stuff. But one of the most common DNA mutations is called a point mutation. And that's when one letter in that DNA sequence has changed. Using CRISPR, you can easily correct point mutations, and that's a huge benefit because there are certain diseases in plants and animals that are caused by just one letter being off.
And you might be able to correct that.
Right.
All right. And at the moment, this is somewhat limited in the size of the material you can change.
Yes, as far as the template goes. Now, if you want to let the cell repair itself, that's fine. As far as providing a template, for right now, we have a size limit. Yes.
Okay. And then there's this other thing that's kind of like a shotgun or something, which sounds awfully crude for something this scientific, but let's talk about that one.
It's very sci-fi, and this is called particle bombardment, or the gene gun. And so with the gene gun is loaded with these microscopic gold beads, pure gold. We coat these beads with our DNA of interest. And then we physically, with brute force, shoot these beads into the plant cells to have them uptake this DNA.
GMO is not a term that scientists like to use.
Right.
How is it understood by the general public, and how do scientists want us to consider it?
Okay, so the term GMO, genetically modified organism, is something that arose in popular media. The public thinks of a GMO as a transgenic organism. A scientist or a breeder or any geneticist, they don't like the term GMO because to them, it's a very broad, vague term. They understand the complexities and the differences between something like GMO or cisgenic or transgenic. And so they don't like to use GMO. When you tell a scientist this is a GMO, their argument is, well, everything's GMO. Because the word GMO, genetically modified, just means that a change has occurred in a DNA sequence. That's all.
Just like I'm a change from my parents.
Right, and so it doesn't mean that the organism, again, to the scientific community, GMO does not mean that the organism is transgenic. It just means that some modification occurred in the DNA sequence.
Well, then let's talk about transgenic and cisgenic.
So a transgenic organism, scientists like to use the word transgenic because it's highly specific. It immediately tells you this organism in question contains genes from an unrelated species, a sexually incompatible species. So I couldn't cross a cat and a dog naturally, but if I were using transgenic technology, I could move genes from the cat into the dog.
And that was done when you moved the glowing genes from the jellyfish into the tobacco.
Right, and so that would be a transgenic organism because it contains genes from a completely unrelated species.
Now, what is cisgenic?
Cisgenic is sort of similar, but instead of using an unrelated species, we use a related species. So if I had a Honeycrisp apple, I love the taste, I love the texture, everything about it. I'm not so fond about how it grows. But I am fond about how Granny Smith grows. I can take a gene from Granny Smith apple and put it into Honeycrisp. Now they're both apples. I could cross them naturally. I could cross-pollination. Or I could take a shortcut and remove exactly what I want and put it into the Honeycrisp. That's a cisgenic change. So cisgenic means that it was created through biotechnology. However, it does not contain genes from an unrelated species.
Jonathan, this is a fascinating topic, and we hear more about it every day.
Yes.
Thank you for giving us the basic building blocks of the vocabulary so that we can start to be a little more scientific in our approach to understanding.
Thank you for having me.